Today I'm going to explain the production of amylase, production and analysis of amylase. Well, the topics out there are, I mean, yeah, the topics. This is the first is the aim. That is the production analysis of amylase. Then it's the introduction, or the process or the principle. Here we're going to learn about. Why we have to isolate, why do we need to isolate amylase, or on what basis, or what principle we're going to do that, and after that the procedure, how we are going to do that. Oh, sorry, now then there's the requirements, what materials we need, and then there is the procedure, and then what is there oh then I guess observation, and then calculation, and then the inference. Well, aim, I already said that it's the production analysis of amylase. Then the principle. Well, introduction. First, the introduction or the theory. Well, um, amylase are a variety of types. One is alpha amylase, and there is beta amylase. Then there is glucoamylase. Then this one is found in humans. It is produced in humans. But all these three types of amylase breaks the 1,4 alpha glycosidic bond in starch. Actually, amylase, you see the amylase, what does it mean? That it degrades amylose, and starch is made up of amylose and amylopectin, I think. um, so when amylose is degraded, so indirectly, starch is also degraded, and it finds its uses in food industry, pharmaceutical, then textile. Now why textile and this? Like food. So we get our food from plants, right? So, and plants, to degrade the starch in them, we might use, I don't know why, but I'm just saying how you can remember this. And then pharmaceutical, like different herbal medicines are produced from different plants, so if there is a need to degrade the starch. And then in textile, like cotton is also a product from plants, and this amylase is produced by humans in the salivary gland, and in their, from their pancreas, and then different bacteria and yeah, plants, they also produce this, and fungi too. And then I would go for the principle, that is on what basis, this product, this thing is going to happen. Actually, what we are going to do out here is, we are going to find out alpha amylase. Now we are going now what alpha amylase does is, it breaks down starch into, if we apply alpha amylase to starch, the starch breaks down into maltose plus isomaltose. So this is it. Now one thing, so when we are finding out, when we are isolating amylase, then how can we find out how much amylase is being isolated? Well, yes, for on what what concentration of amylase is being isolated. Now what we can't do is, we can't, if we estimate the amount of protein, like one doesn't BSA Lowry method, in that one, if we try to, find out the amount of protein in, then no the solution might contain different other proteins along with amylase plus other proteins. So the problem is it that when we are using this method, we are also counting the, the concentration of protein as the concentration of amylase. So that's the problem, right? So we only need to find the concentration of amylase. So how can I do that? We know that amylase, amylase converts starch to amylose, right? If most amylase is produced, then within the same time, then more starch will be converted to maltose if enough substrate is there. That is, if enough starch is there. So, if we find out that, if we can find out the amount of maltose, yes, after a given time, after it is made allowed, after that amylase enzyme is allowed to degrade starch, if we find out the amount of maltose, actually I'm talking about the analysis part, okay. If we find out the amount of maltose, then what we are getting from it, we are getting that how what is the quality of that amylase, like we are getting the we are getting the, what is that called, activity of the amylase, sorry, yes, of the amylase enzyme. If we calculate the amount of maltose in the solution after a given time, um, after a specific given time of, when the amylase when the amylase is allowed with a with a given time to degrade the starch, then the amount of maltose produced specify, tells about the how much, what is the activity, or what amount of activity that enzyme is showing. Well, this is it. And one thing, what is activity of an enzyme? First of all, you need to know that, right? Well, activity. Well, it is the amount of substrate produced per time in a given time. Like what, for the, like this maltase is this, sorry, this amylase is given this much time, t time, and it produces p product, then the activity of amylase enzyme is p by t, okay. So the unit of activity, the dimensions of activity is like this. um yeah, that's it. And and this enzyme activity is, this enzyme activity, its units are like cattle. There are many units of it. One is cattle and one is u. Well, one cattle is equal to 1 mole per second, okay, and one u is equal to 1 micromole per minute, okay. Well, you can remember this, that this one u is equal to 1 micromole, because mu actually looks like quite u, right? So I mean, one u is equal to 1 micromole, because mu looks quite like, um, mu is quite similar to u in looking, right? That's why, um, that is 1 mu per mole, 1 mu mole per min. Now, how you can remember that 1 mu mole is equal to, a one, when it is 1 mu mole per, when it is 1 mu mole, when it is mu mole, then it is per minute. Well, for that you can remember it like this, like this mu also looks quite like, it is quite similar to m also. And more, and other than that, you can remember it in this way, that if the activity is very low, then this is used, then u is used. That's why, like, after when 1 minute is crossing, then we are we are calculating in mu mole, and here in when 1 second is coming, even then we are considering in 1 mole, in mole, sorry, not 1 mole, in mole. So one cattle is for bigger, when is used when the specific, the activity is high, when the activity of the enzyme, and this is used when it is quite lower. And the relationship between one u and cattle and Cattle, yes. Is this like 1 U is equal to 1 micromole, right, per minute. So it is 1 by 10 to the power 6 mole per minute, or equal to 1 to the power this again, and then per second, then yes, then this is going to be 60 or what. Wait a second. Oh no, it is going to be like this into 60. So it is like 6 into 10 to the power 7, I mean, on the denominator side of cattle. Or if we have to convert it into nano cattle, then we can do 10 to the power 9 by 6 into 10 to the power 7 in cattle, and then 10 to the power, I mean, 6 nano cattle, and this is equal to 50 by 3 and get r 1 6 7. This is 16.67 nano cattle. This is it. And then, well, now the question is how we can calculate. Now I said you that if we calculate the amount of concentration of the maltose, then um, we can do it, right? Now the thing is like how we are going to calculate the activity of the enzyme. As I already said that I have said, but we have to make a like wait, let me think. So we have to make a so we want to calculate the concentration of maltose, right? So but before that, before knowing these, we have to know the concentration at all. I mean, we have to know the how we are going to know it by using photospectrometer. Okay, but why will it give a specific color? Well, for having a specific color, we use DNase. We know that maltose is a reducing sugar, right? And DNase is 3,5 DNase, that is D,5 dinitrosalicylic acid. Okay, but what is like, I mean, I don't actually remember the structure of, yeah, salicylic acid, but nitro is like this, right? I guess so. I don't remember these two well. Yes, and so this is present at 3 and 5 positions of salicylic acid, this nitro, 2 nitro. So when one is being reduced, that nitro at 3 position is being reduced, so it becomes amine. So 3 amine, 5 nitro, salicylic acid. So that, and it has a sort of deep red type of color, and it is quite orange in color, and its maximum absorbance is at 540, yeah, nanometer, and that's it. So first of all, what do we need to know? I mean, what do we need to do? Well, first of all, we need I mean, for what OD, what will be the maltose? How would we know that? For that, we need to do experiment. At what OD, what is the concentration? So we need to make a standard curve. Okay, I'm just telling out here the procedure in short or briefly of the analysis part, obviously. And so at first we need to make the standard curve, make standard graph or curve, and then find the slope of it, because we know that in Lambert-Beer law it will be a straight line, right? The OD versus concentration curve or graph, and find the slope. So if we find the slope, then on getting out, I mean, any concentration, on finding any OD, we can find out the concentration of the maltose. even if we haven't tested for that one because it's a straight line. And so out here, if OD is here, I mean in here, and this is the, yeah, what is it called, concentration of the maltose. So we have, so it is like y is equal to m is that slope mx, and mx is this one. So after that we are, I mean we can find out this one, right? I mean we have got y when we are finding out the OD for something, OD for the unknown concentration. So on dividing this one, this OD by m, or the, what is it called, by the slope, then we can find out the that concentration of the unknown concentration. We can know the unknown concentration of maltose, right? Well, that's it. And then since we know how much maltose is I mean since we know the concentration of maltose, so we can find out how much maltose is produced by multiplying it with the volume, right? So if we know that one, that how much maltose is produced, from that, by dividing it with the time, we can actually calculate the, um, what is called, we can calculate the, activity, right? So that's it. And what more, so next we need to do it in quite detail. Now, what are the, I mean, materials needed? Um, well, I'm seeing and saying you. Well, the materials needed are, um, well, I'm showing it. That's better, I guess. Well, let me cover my pen, else I will be marking it. Well, here it is. Array camera. Wait a second, please. Oh, well, here it is. So here, what are the things written? Well, let's start from the beginning. So at the beginning, I mean, for the, I mean, for the isolation, or the, for the, yes, for the isolation of, well, I have, well, for the isolation of the, yeah, what is that, amylase from that. Well, let me tell you one thing. After we have screened the bacteria, you know that we need to screen the bacteria out using that iodine solution and all that, using the starch media and all that, as we have learned in the, or we have got to know in the previous video or in the previous practical. Well, so after that, what would we be doing? Well, we would be culturing that bacteria, or allowing that bacteria to ferment in a fermentation media. So we need a fermentation media, right? For that, we need to prepare it. Well, we can consider it later. We can consider it in the procedure section. But here, what we need to consider is that the requirements. So after that one, we need a centrifuge, right? Although it's not written out here, we need a centrifuge to get the supernatant, which contains the, what is it, the maltose, sorry, the amylase. And actually, what I have written out, actually I've done a mistake actually in this one, that I have written that the amylase is being, actually in that research paper I found it. That's why I did a mistake. By mistake, I did the mistake that the amylase was being isolated or extracted by centrifugation. But actually, what I found out later is that, yeah, that the supernatant was being collected, right? The cells were being pelleted out, so we are getting the supernatant. But that supernatant is not, is actually, it also can, yeah, along with the amylase, it also contains some other impurities. And so ammonium, I guess sulfate is added. I've forgotten, by the way. Ammonium sulfate or something is added, which precipitates out the protein, and then that precipitate is taken out, and somehow from them that actually that amylase is being is extracted. I don't remember that one. Actually, this is what happens. um that's it. And but that also might contain other proteins, right? But so that how the amylase is produced from other proteins, I don't know. Well, so it is it. And what more, I don't remember. And oh, I was talking about the materials, right? So we need what after that? We need the, I mean, the as to make a standard cup, we need the maltose stock solution. Here it is written that the concentration is 1 gram per microliter of 50 ml volume. So here the, I mean, secondly it is written the amylase. Obviously we need that to test amylase, but here it is written as 0.1%, and I think that is actually wrong. I have originally made this, but I think that there is some problem out here. So let's skip that. Let's pull the examiner. That's it. And then the starch solution, and then the DNAs, which using which we are going to detect the amount of maltose. Then conical flasks, pipette, test tube. Let's not do it in this way. So let's think it out using by the procedure. So first of all, um, we obviously need the amylase, right? And then the maltose stock solution to make the standard curve, and then for testing the amylase, we need, for that particular sample, we need starch solution, then the DNAs, as we know. But in the way before that, while fermentation, we need the conical flask, pipette, test tube, measuring cylinder, distilled water, colorimeter for the testing, and boiling water bath for stopping the, I mean, after the DNS is added. After that, boiling water bath, cuvette for the colorimeter, I mean for the photospectrometric analysis, and buffer. Well, that's it. And here the procedure. Well, one thing, these procedures and the preparation methods, I'm actually not going to go into much details now, or because later on I might be making videos using tricks that would help you to remember this or memorize these, the composition of these. I need to say that. Well, first of all, what do we need? First of all, we need to need a fermenting media, right? So here is the composition of the fermenting media. Well, wait a minute, I don't think it's focused. I don't know why it is not getting focused. What is your problem? Oh yeah, now it's focused. Yeah. And first of all, the fermentation solution. So one thing, it must contain starch, right? Well, it needs a protein, some sort of, we know the starch is a carbohydrate, so peptones. Then for the nitrogen, here ammonium sulfate. For, and here are buffers. I mean, we know that phosphate is a sort of buffer, and potassium for this is there, and then magnesium chloride. These are there. What am I doing out here, man? And then we need the maltose stock solution. Actually, those part which you have to memorize, I'm going to say later. Okay, maybe in a different video. I don't think it's much of focus, right? It is so much blur type. Well, that's it. Well, well, so we need to prepare the maltose stock solution for getting the OD at different concentrations of. I mean, we have to prepare the stock solution, and that's it. And then we need to. Well, actually, what it is written out here, I mean, it should actually be, I mean, in out here, in the requirements, it should actually be 10. Because initially, actually, it was written 1000, I mean, the derivation was 10 down to 1000. Okay. That's why we took, I mean, that's why it was written 0.1. Actually, it should be 10, because here I have taken 10, 10-fold dilution. Okay. And that is it then. Well. Preparation of DNS. Oh, then what do we need to do? We need to prepare the DNS, and DNS is not that soluble in water, and for that we need to use NaOH and this. You see this? I can't say anything. So we have to prepare the DNS, and after that the start solution, which would be used in the which would be used um to which would be used to get degraded using the amylase for testing out its activity. And then what do we need to do? Well, we need to do is the preparation of the maltose. So so we need to switch on the spectrophotometer 15 minutes before doing this. Well, so we'd be taking maltose and water in different, I mean, amounts, but the total amount would be the same. To vary the concentration of the maltose, but to keep the volume same. And add 3 ml of DNS to all of those test tubes, and the OD would be acid 540 nanometer. Well, here, see out here. So here is 0, so this is going to be the blank, and then there is 0.2, that means, and there is the total volume is 1. So this is, I mean, 20% solution of maltose, this is 40%, 60%, 80%, and this is the 100% to create the standard curve. And what more, and this is the standard curve. Then here we have calculated the what is called the slope from the curve. Here is it is. And so So here the height is 0.34 and the width is 0.26. So here it is, and then after, wait a sec please. So here is the production. It is written here it is written that after doing the screening test, the bacteria were subcultured at 4 degrees Celsius, and now from this culture, from that, what is it, from that culture, it was transferred to a fermentation medium. To that fermentation medium, I mean from that there was this screening. Okay, this, that petri dish in which that screening was done. From that, it was transferred to another. Those bacteria transferred to another and to another agar media containing the starch nutrient, and from that it was transferred to another starch nutrient broth. Okay, the liquid media, and it was incubated at 40 degrees Celsius, at 120 rpm for 24 hours. Okay, I don't remember. I'm just seeing and saying. And then fermentation medium is inoculated with this. This liquid thing. This liquid media was added to the fermentation medium. Okay, so that liquid media was 20% of the total formed out here. Total fermentation media formed out here, and it was shaken at 37 degrees Celsius for 24 hours. And after the fermentation period, the culture medium was centrifuged at 10,000 rpm. Oh, you can't see, at 10,000 rpm, and the supernatant was extracted. This is the this is the enzyme, but as I said you that actually ammonium sulfate is used. And after that, for the purpose of what is it, analysis, here it is written. Actually, this has been done to the amylase is diluted. Here 10-fold dilution is being shown. Actually, this dilution is done to keep the OD between 0.1 and 1 in such a way, and then that amylase is allowed to degrade starch. Here these things are written: 1.5 ml of 1% starch into and buffer solution, and to maintain the pH, and this. So that reaction was done, and then to check the maltose produced, spectrophotometric analysis was done. So the optical density was obtained. That is it. And then now the calculation part. So I have said you that how to calculate the concentration of the maltose beforehand, that by dividing this on this. Now the question is that I think I should make another video, because this video is getting quite bigger, and after that I have to make another video on the topics, on the tricks to remember, how to remember this. In just the next video I'm going to tell you about that how this formula is coming out, all that. Well,